You know, your body is fighting invisible wars every single second of every day. But how do you actually watch a battle you can't see? Well, today we're going to pull back the curtain and look at the incredibly clever ways scientists make the microscopic world of your immune system totally visible. I mean, really think about that for a second. We've got this amazingly complex army of cells and proteins inside us, but it's all happening on a scale we can't even imagine. So how in the world can a doctor know what's going on in there? How can they count the soldiers, check the weapons, and figure out who's winning? That's the huge challenge we're diving into. And the secret, the key that unlocks almost everything, comes down to one beautiful, super specific interaction. It's the bond between an antibody and its target, the antigen. Think of it like a perfect lock and key. The antibody is shaped to fit only its specific target, and pretty much every technique we're about to see is just a different, clever way of spotting that exact moment of connection. Okay, so let's kick things off with a really common problem. What if the thing you're looking for, that antibody or antigen, is there in such tiny amounts that it's practically impossible to find? You know, it's like trying to find one specific grain of sand on an entire beach. The solution here is just so smart. Instead of searching for that one tiny thing, scientists realized they could look for the evidence of a fight. See, when an antibody finds its target, it can trigger this massive biological chain reaction, and that reaction is way, way easier to spot than the first little event. It's kind of like looking for the giant plume of smoke instead of trying to find the tiny spark that started the fire. And a classic example of this is the complement fixation test. Okay, so here's the play-by-play. First, you take the patient's sample and you mix it with the antigen you're testing for. If the patient has antibodies for it, they'll grab on. Then you add a very specific amount of that chain reaction fuel we talked about. It's called complement. And for the final step, you toss in some indicator cells, in this case, red blood cells, which are basically bait for any leftover complement. What happens to these red blood cells tells you everything. And here is the really cool, kind of backward part of this test. Just look at the positive results column. If the patient's antibody was there, it formed a complex that used up or fixed all the complement. So when the red blood cells are added, nothing happens. They're perfectly fine. So no destruction actually means a positive result. On the flip side, if the antibody was missing, all that complement is just floating around, free to attack and burst the indicator cells. And that's your negative test. So clever. All right, so knowing if an antibody is there is one thing. But sometimes you need to know more. You need to know how many of your cells are actually busy making that specific weapon. What's the real strength of your immune response? I mean, think about how crucial this is after something like a vaccine. The question isn't just, did it work? It's how well did it work? How big of an army did your body actually build? We need a way to do a head count of our cellular soldiers, one by one. And the go-to technique for this is called the E-Ellis spot assay. And it's pretty much what it sounds like. It literally creates a visible spot for every single cell that's pumping out the protein you're looking for. It's like forcing each tiny soldier to leave a visible fingerprint behind. So here's how they do it. You start with a special plate that's been coated with the target antigen. Then you add the immune cells from your sample. Now any cell that's making the right antibody will start releasing it, and that antibody gets captured right there on the plate. Next, you add a second antibody that's linked to an enzyme. And for the final step, you add a chemical that reacts with that enzyme, and boom, it creates a little permanent, colored spot right where that one active cell was setting. And this is what makes it so incredibly powerful. You don't just get a vague signal like strong or weak, you get a hard number. You can literally look under a microscope and count the spots. Not a lot, but exactly 2,341 individual cells doing their job. That level of precision is just a game changer. Okay, so far we've been talking about things that happen mostly in a lab, but now we're going to talk about a situation where these tests have huge real-world consequences. We're talking about those rare, scary times when a mother's own immune system can actually become a danger to her unborn baby. This is the classic problem of Rh incompatibility. Basically, if a mom is Rh negative and her baby is Rh positive, getting that from the dad, her immune system can see the baby's red blood cells as foreign invaders. If that happens, she could start making antibodies that cross the placenta and attack the baby's blood. It's a really dangerous condition. Thankfully, a brilliant immunologist named Robin Coombs developed a test to catch this problem. The Coombs test is designed specifically to find those dangerous antibodies from the mother. It's absolutely critical for spotting the risk early so doctors can step in and protect the baby.
And what's really smart is that there are two versions of the test. The direct Coombs test is done on the baby's blood after birth. It asks, are mom's antibodies already stuck to the baby's red blood cells? But the indirect Coombs test is done on the mother during pregnancy. It's a screening test that asks, does her blood even have these potentially dangerous antibodies in it? Both are so important for keeping babies healthy.
All right, for our last big challenge, we're going to scale way up. The immune system, as you know, isn't just one type of cell. It's this incredibly diverse army of T cells, B cells, killer cells, you name it. So how on earth do you sort through that complex mix and count everything? Well, the solution sounds like it's straight out of science fiction. Seriously, you basically take this jumbled mix of cells, you tag each different type with its own unique fluorescent color, and then you line them up and shoot them, single file, past a laser at a mind-boggling speed. This amazing piece of technology is called flow cytometry. Here's the gist. First, you tag your cells. Maybe you make all the T cells glow green and all the B cells glow red. Then the machine uses fluid to funnel them into a stream so narrow that only one cell can pass the laser at a time. As each cell zips by, detectors measure its size, its complexity, and exactly what color it's glowing. And some of these machines can even physically sort the cells into different collection tubes. It's incredible. And this is what you get at the end of it all. This beautiful, crystal clear breakdown of a once messy sample. You know with absolute precision that in this sample, you've got 65% T cells, 20% B cells, and so on. This kind of data is absolutely essential for diagnosing diseases, checking if treatments are working, and doing all kinds of vital research. So when you step back and look at it, it's pretty amazing. We've seen how immunologists solved these totally different problems. They found ways to detect tiny invaders using chain reactions. They figured out how to count single cells by making them leave a little spot. They learned how to protect newborn babies from an immune mismatch, and they can sort an entire army of cells using lasers and light. Ultimately, every single one of these tests is about one thing: making the invisible visible. They take these silent, microscopic battles happening inside of us and turn them into clear data we can actually understand and use. And that ability to see is what changes medicine from guesswork to precision. It really just makes you wonder, what other invisible worlds are we going to learn to see next?